Hello, my name is Mohammad. Today I will present my project on design and validation of a p53 expression factor. The aim of this project is to express human p53 in mammalian cell to study its role in cell cycle arrest and apoptosis. P53 is known as the guardian of the genome, responding to DNA damage by stopping cell division. I use Uniport code P04637 for T53, which encoded 393 amino acid protein with essential DNA binding and tetramerization domain. This is the amino acid sequence and reverse translation of p53. To express p53 in mammalian cell, I select expression vector pcDNA 3.1 because of it has a strong CMV promoter, antibiotic resistance marker, and a multiple cloning site like HIND3 and XBAL restriction site. For proper expression, I design my gene like that way. At the 5' end, I add HIND3 restriction site codec sequence and stop codon ATG. And 3' end, I added 6 hitch tag for protein detection and purification, a stop codon and XBile restriction site. The total insert size is approximately like 1.2 kilobytes. The design ensure correct reading frame and efficient protein production. This is my final recombinant plasmid containing CMP promoter, 6 histag, BHA polyadenylation signal, ampicillin resistance site, neomycin resistance, and the HIND3 and XBile fragment. The next step was bacterial transformation. I introduced the ligate plasmid into competent E. coli DH5 alpha cell using a heat shock at 42 degrees Celsius. The cell was recovered into SOC medium and pellet on LB ampicillin pellet to select bacteria carrying the recombinant plasmid. To confirm successful cloning, I performed colony PCR using CMV forward and BGH reverse primer, which produced a 1.4 kilobytes band. I then isolate the plasmid by mini-prep and verified it by restriction digestion using HIND3 and XBile. Finally, Sanger sequence confirmed the correct p53 sequence and reading frame. To confirm plasmid was transfected into mammalian cells such as HEK293T, a p53 null cell. Protein expression was checked by Western blot using an anti-Hig antibody. I expected a band at around 53 kilodalton. To test my function, I analyzed expression of p53 target gene like p21, cell cycle arrest, apoptosis. I could also purify the protein using NI anti-chromatography because of the Hig tag. In conclusion, I successfully designed and validation at a p53 expression vector. This system allowed me to study how p53 control the cell cycle and apoptosis, which is very important in cancer research. And here is all of my references. Thank you, everyone.